Hello, everyone. My name is Justin, and today the subject of my presentation is on Ebola virus disease, or EVD. Now, before I start, I want to clarify that there are several known viral species of Ebola virus that causes EVD. So, for the purpose of my presentation, I will be focusing on the Zaire Ebola virus strain. Now, the Zaire Ebola virus belongs to the family Filoviridae, which is a family of viruses that are single-stranded and whose genome is RNA. The size of the virus is approximately 970 nanometers in length and 80 nanometers in diameter. Its morphology is cylindrical or tubular in shape, as you can see in the picture on the right, and it does not have an arrangement as this is applicable to bacteria, not viruses. And since it is a virus, it has a capsule, specifically a helical nucleocapsid composed of several types of proteins that protects the RNA genome. Now, surrounding the Zaire Ebola virus is an outer envelope that is derived from a host cell. And one important thing to mention is that the envelope is studded with glycoproteins. Now, I will discuss this particular point in more detail in a later section. Now for the epidemiology of Ebola. Ebola was first spotted in 1976 in the Republic of Zaire, now called the Democratic Republic of Congo. This eventually turned into an outbreak in the surrounding region, as you can see in the image on the right. Now, there were additional outbreaks later, such as the most recent outbreak from March 2014 to December 2016. And this is the time period that my presentation will be focusing on. So, some relative facts about Ebola is that the name originates from a river in Africa where it was first spotted, called the Ebola River. And as for the Zaire Ebola virus in particular, it has the highest mortality rate out of all species of Ebola virus. This mortality rate was reported to be between 60 to 90%. And the reason for such a high mortality rate is due to poor health infrastructure and no proper treatment or vaccine at the time of the outbreak. Now, I want to clarify that I was unable to find data for both incidence and prevalence because I was using data from years ago. However, I do have additional data to make up for this in terms of how many cases of Ebola were suspected, probable, or confirmed during the 2014 outbreak. Now, there were a total of 28,652 cases, and these cases included the countries of Guinea, Liberia, Sierra Leone, Italy, Mali, Nigeria, Senegal, Spain, the United Kingdom, and the United States. And including all the countries mentioned right now, there were a total of 11,324 deaths as a result of Ebola. Now, I will be talking about the people at risk of Ebola based on age group. Now, 43% of children less than 2 years old were infected, 30% of children between the ages of 5 to 14 were infected, and more than 60% of adults ages 30 and above were infected. So, based on this data, adults were more likely to get Ebola than young children. Now, the place of interest for Ebola is sub-Saharan Africa, and this is because what I mentioned before in the previous slide, in that the first outbreak originated in Zaire in 1976. Now for the virulence of Ebola virus. When Ebola enters the bloodstream, it can inhibit the host's defense mechanism, more specifically the immune system. It does this by first attacking macrophages and dendritic cells. And when these cells become infected, they produce pro-inflammatory cytokines, which are important for downstream immune response. But because there are so many cytokines being released at once, this causes contradictory signaling, which interferes with proper immune response. For example, some cytokines would increase vascular permeability, leading to leaky blood vessels, and other cytokines would cause coagulation or blood clotting. The effects of these will be explained in a later slide. Furthermore, the infection of macrophages and dendritic cells prevents T cells from being activated, which hinders innate immunity as well as adaptive immunity, since T cells wouldn't activate, so they wouldn't be able to aid in antibody production later down the line. Lastly, natural killer and T cells would also be targeted by Ebola virus, leading to apoptosis, which is programmed cell death. Both of these cells play a vital role in immune response, so ultimately Ebola virus would be severely hindering the immune system, which can lead to more complications down the line. Now, as for how Ebola virus gains nutrition from a host, this has to do with the glycoproteins attached to the Ebola virus envelope. When the glycoprotein binds to a receptor on a host cell, this triggers macropinocytosis. What this means is that the host cell would engulf the Ebola virus along with the surrounding fluid nearby. The image on the right here demonstrates this happening. Once this occurs, the Ebola virus would eject its viral RNA into the cytoplasm of the host cell. Then the Ebola virus would then essentially hijack the host cell's proteins so that it can replicate more copies of itself in terms of more viral RNA and proteins. And once the new viral particles are fully assembled, the completed Ebola virus particles would either bud off from the host cell or rupture the host cell entirely. Both methods would release viral Ebola particles, which would then lead to other host cells becoming infected. Now for the etiology and transmission of EBD. The reservoir of Ebola is infected animals and humans. Examples of infected animals includes monkeys, chimpanzees, and fruit bats. It should be noted that dead humans and animals who were infected with Ebola can still serve as a reservoir. Now for the mode of transmission for Ebola. It is through direct contact with blood or bodily fluids. Examples of this includes vomit, diarrhea, urine, breast milk, sweat, and semen. Now, one thing I want to clarify is that Ebola does not spread through the air, water, or by food. However, the only exception in which it could spread through food is if the food originated from an infected wild animal. This is referred to as bushmeat. By preparing or eating bushmeat, you are more likely to come in contact with the blood or other bodily fluids of the infected animal. And remember that Ebola can spread from the dead carcasses of infected animals. As I mentioned before, Ebola targets the immune system. In particular, they target dendritic cells. And because dendritic cells get infected, they are unable to activate T cells. Now, T cells play a role in neutralizing the virus and infected cells, as well as aiding in the production of antibodies. Now, because the immune system is drastically weakened in this case, the Ebola virus can wreak havoc in the body without much interference from the immune system. In addition to the immune system, Ebola targets the liver. It specifically wipes out cells that are required for producing coagulation proteins and other important components of blood plasma. Also, Ebola targets the adrenal gland. So, because of this, it cripples cells that make steroids, which are very important for blood pressure regulation. Now, I will come back to this in a later slide by discussing the complications that arise from all this. Now for the physiological and medical symptoms of Ebola. I will first start by discussing the signs. A person who has Ebola may experience severe headaches, muscle pain, weakness, fatigue, loss of appetite, diarrhea, and vomiting. As for the symptoms, one that can be measured by medical personnel is whether they have a fever. And according to the CDC, fever is a body temperature that exceeds 38 degrees Celsius. Now, the problem with the signs and symptoms that I just mentioned is that a patient may be mistaken for having another disease, such as Marburg virus or malaria, since they also have similar signs and symptoms. As such, to confirm whether or not a patient actually has Ebola, a reverse transcriptase polymerase chain reaction test can be performed. This test can be abbreviated as RT-PCR. How it works is that a blood sample will be obtained from the patient. The Ebola RNA will be purified in order to isolate it from the patient's blood sample. Then, selective primers that match that of Ebola RNA would be added so that it can attach to it. Then, reverse transcriptase enzyme would be added, which would synthesize a complementary DNA strand of Ebola. After, the application of heat as well as Taq polymerase would be utilized in order to amplify the amount of Ebola DNA virus particles. Once all is said and done, the sample would be analyzed for the presence of Ebola virus particles. And had the patient not been infected with Ebola from the beginning, there would not be any virus particles present after performing all these steps. However, if the patient was infected with Ebola, there would be virus particles present in the sample. Therefore, it can be determined that the patient is in fact positive for Ebola. So this is how the reverse transcriptase PCR test can be utilized in order to diagnose a patient that is suspected of having Ebola. Now for the complications of Ebola. As I mentioned before, Ebola virus targets immune cells such as macrophages and dendritic cells. This would trigger the release of cytokines that shouldn't be released in this situation. Some of these cytokines would induce coagulation, which is the formation of blood clots in the bloodstream. When blood clots form, this blocks blood flow to organs such as the liver and adrenal gland. So, when not enough oxygen is given to the liver cells, they start to die. And this is a problem because liver cells are important for inducing coagulation in the bloodstream. So, without liver cells, this increases a patient's susceptibility for hemorrhage, which is when blood vessels rupture. In addition, loss of oxygen to the adrenal gland affects the production of blood pressure steroids. So if the adrenal gland is not given enough oxygen, it cannot produce these steroids. As such, the body would not be able to control blood pressure, leading to a drastic reduction in blood pressure. Ultimately, it would be the case that the patient dies from a combination of low blood pressure, multiple organ failure, and shock from infection. An infection can happen since the immune system was severely compromised due to Ebola. Now for prevention and treatment. In order to prevent possibly contracting and spreading Ebola, this is what can be done. Washing your hands frequently using soap and water. Make sure that fruits and vegetables are washed and peeled thoroughly before eating them. This is to ensure that any blood or bodily fluids containing Ebola are eradicated. Avoid physical contact with people who has possible symptoms of infection. Do not handle dead animals or raw meat. Remember that Ebola can spread from dead animals that are infected from Ebola, especially their blood. Lastly, do not eat bushmeat. As I said before, bushmeat is meat that comes from a wild animal. It may or may not be infected with Ebola, so be cautious of this. Now, for treating the symptoms of Ebola. During the onset of the outbreak, there were no licensed treatment or vaccine at the time. As such, medical personnel managed the symptoms by doing the following: giving fluids and electrolytes, supplemental oxygen, blood pressure medication, and blood transfusions. This ties back to the previous slide that mentioned that a complication of Ebola is reduced blood pressure and oxygen levels. Now for a drug that could help with treating Ebola. The name of the drug is Abanga, and it is a recombinant human immunoglobulin G1 monoclonal antibody treatment. It was recently approved by the FDA on December 2020. Now for the mechanism of the drug. It is a form of antibody treatment, and the antibodies were isolated from a human survivor of the 1996 Ebola outbreak in Zaire. It is administered as a single 50 milligram IV infusion over 60 minutes or as a single dose of 400 milligrams. Now, the antibody targets the Ebola virus surface glycoprotein. And as I have previously mentioned, the glycoprotein, which is embedded on the envelope of the Ebola virus, plays a key role in how it enters host cells. So, given that Abanga antibody binds to the core of the Ebola glycoprotein, this blocks the Ebola virus interaction with the receptors on host cells. So with this treatment, it helps to block the ability of Ebola virus from entering host cells, thus impeding the spread of the infection. And when this treatment was implemented in the Democratic Republic of Congo, it experienced an 89% survival rating, which demonstrates that the treatment does work. Now, this slide and the next one are for bibliography purposes, in terms of citing the sources that I used for my presentation. And this concludes my presentation. Thank you for listening and have a great rest of your day or evening.